You're just going to cut the two hemispheres in half. You kind of want to make it like a clamshell, though. You don't really want to cut it all the way through because the pituitary gland is really difficult to cut all the way through, so you lose the pituitary gland and infundibulum if you go all the way through. So cut it down to about here, and then you can identify all of the structures internally. Also, with that said, we have some brains. I spent some time in the earlier class going through and picking out brains that had already been dissected that are like pretty good dissection models. So when I give you your brain, you can decide whether you want to do the longitudinal cut or whether you would like to get one that has already been cut. All right, so you can decide that with your group, but just know that's an option. Okay, so I'm going to point out the mammillary body and peduncles now and the colliculi from the internal view now. So, actually, hang on. I want to jump back just to make sure I was telling you guys correctly. What did I say this was? Pineal gland? Okay, I just want to make sure I told you right. That is pineal gland, but as I was looking at this one and reading mammillary body, I was like, oh, I'm killing myself. I told you the wrong thing. All right, special view. Okay, so we'll go over the mammillary body first. Just to reorient you, here's our pituitary gland right here. This would be a cross-sectional view of the optic chasm. And here's our olfactory bulb. So those three are kind of right in a row, right beneath there. Directly above the pituitary gland, we have the mammillary body. So you're just going to look right up above it. Then for our colliculi, you'll look just in front of the cerebellum for these two bumps here. And you'll notice that they do differ in shape in the sheep brains. The inferior colliculi is a little bit thinner, and it also will appear to be more white, whereas the superior colliculus is more rounded and like a more yellow color, relatively speaking. Okay, now we'll move on to the pons. The pons is going to be down by the pituitary gland. It's this oval shape right here. Notice it's a lot smaller than the pons in humans. All right, and then the oblongata, or the medulla oblongata, will be just below that. All right, so orientation-wise, it'll be just like the human brain. All right, now for our ventricles. So first let's start with our lateral ventricle. That's going to be right in here, just like we saw last time. Now, when you get your brains, regardless of whether you want to dissect or not, what often happens is that one side of the dissected brain will have a sheet covering the lateral ventricle. That will be the septum pellucidum, okay? So one side usually has an intact septum pellucidum, and the other side will have a simply open lateral ventricle. So don't expect it to be like perfectly symmetrical in that sense. Now the third ventricle is simply going to be the space here taken up by the thalamus and the hypothalamus. So again, for that, we just put a pin anywhere in this area and ask you to identify the ventricle or the space in that area. The aqueduct is going to run right underneath the inferior colliculus, right down here, right underneath the cerebellum to here. Once we get to this area here, we're in the fourth ventricle region. So that would be a fourth ventricle right there. Now for the choroid plexus, we found it on the models right here, was that pink piece of tissue in the brain models right there. But in sheep models, you actually can find the choroid plexus in a lot of places. Like, I guess that's true for human brains too, but you kind of have to dig for it. The choroid plexus, remember, was the network of capillaries and arteries. So you can see this dark structure right here. Can everyone see how it's kind of like almost a blue color right in this region? That's because there's a choroid plexus in there. So in order to find the choroid plexus, the best thing to do is to take one of the blue pointers we have, and right here, specifically in the third ventricle, just run the poker through this area and lift up, and you'll usually come out with something that looks like a blue string, okay? And that's your capillaries and arteries, the choroid plexus. So you're going to look right underneath the fornix and right above the thalamus in the third ventricle to find that. All right, so next one we're doing the corpus callosum. The corpus callosum is this white strip right here. So it's the most superior structure that we really discuss in this middle brain area. And flip the page, we only have a few left. Next we have the fornix. The fornix is kind of like the bottom or inferior border to that lateral ventricle. So the corpus callosum comes up and around. The fornix sweeps down underneath, but they do look very similar. Now the massa intermedia in sheep is actually very different than in human brains. Remember, the massa intermedia was just a small dot on the thalamus in the human brain, but here in the sheep brain it actually takes up the entire area. So once this is bisected like this, in order to find the massa intermedia versus the thalamus, basically what you have to do is take the brain and kind of tip it in the light, and you'll see that some area of this circle is going to be shiny, and some area is going to be rough. The area that's rough is rough because it was cut. So it was actually a connection point that went all the way through the two hemispheres that you cut. So the rough part that you see will be the massa intermedia, and the shiny part surrounding it will be the thalamus, okay? The massa intermedia is about half a centimeter to a centimeter in diameter, so it's pretty small, but it will take up approximately, I'd say at least 2/3 to 3/4 of this flat surface here. Now for the exam, this is really important. Because the massa intermedia takes up so much of the thalamus, we can't actually correctly pin the massa intermedia versus the thalamus and ask you to identify one versus the other, because it's really hard to get that iridescence to show when you can't touch the brain. So for the sake of the exam, if we pin that region, we would accept, or I would accept, either massa intermedia or thalamus, because it is very difficult to tell those two apart, okay? So one and the same on the sheep models, on the actual brain human models that we have, you can tell the difference. So you'll need to know the difference for them. All right, you don't need to know anterior commissure or posterior commissure in the sheep brain, as it says in parentheses, models only. Moving on to the pineal gland. The pineal gland is going to be just anterior to the two colliculus, right here. Now with the pituitary, or the pineal gland, you often will get a brain that's cut, or you will cut in a way in which it only ends up on one half. Okay, so you'll have like the full gland on one half and no gland on the other half. So if you can't find it on one half, just look at the other, you'll probably see it there. Next, the thalamus, again, this region right here, and the hypothalamus would be just below, right here, so hypo for below. Last, we have the arbor vitae. So in the cerebellum, we have the white matter here that looks like a tree. Again, that's the arbor vitae. Now, if we're asking for white matter or gray matter, we would simply pin where it's darker and gray or lighter, like a white color, and ask you to identify the tissue or the matter that it's made of. So if you see a pin here, we're asking for the tissue, gray matter. If you see a pin in the arbor vitae and we're asking for the tissue, we're asking for white matter. Okay? All right. Any questions? All right, so for this